Can you hear me? Bit of a change of gears in the short time in this session. Really all I have here is an ad for what I think is an important area of science. Part of the theme of this program for the 6 months has been new areas generating big data. This is ultimately going to be a really big data generating area. Right now the data is quite poor, but the models involve big parameters, big latent processes. Okay. So those of you in the front can see these bacterial cells going through multiple mitoses over a period of time. So each of those green blobs is a bacterium, E. coli, and the green is fluorescence measuring the level of RNA of one particular gene that's been fluorescently tagged. Okay? And it looks like it's a nice kind of cephalopod at the end there, but it's a colony of cells. I can replay it for you. And it's generating a multivariate time series. Here we go. Each of the cells has a level of the protein that you measure noisily. There's some genetic machinery that's changing that level as you go through time, interacting with other genes. The cell splits, the two daughter cells inherit the protein, et cetera, et cetera. So this is imaging data in the form of movies that is generating multiple time series that is relevant to looking at little genetic interactions between genes in models of genetic architecture. And the specific focus that this field has right now, and it's really only a couple of years since the first paper in this field, the technology is about to explode in order to be able to measure many proteins simultaneously in real time in individual cells. Much of the focus is in the stochastic nature of the way in which proteins interact. Fundamental biochemistry is discrete time stochastic, so cells can look very different. Okay? And that's really the overarching theme, the area that we're interested in. Why bacteria? Because they're easy to engineer, easy to study. You can do this with mammalian cells, you can do it with yeast cells. I'll mention some of that at the end. One of the reasons we're interested in this kind of data is the connection with cancer. Here's a set of genes and a set of arrows that represent some kinds of relationships of these genes in mammalian cell developmental processes central to all cancer. So we have a program of generating this kind of data on mammalian cells of different cancer cell lines to look at how different they are in models that model that kind of data. The bacterial systems are interesting because you can take your favorite little piece of a mammalian network. This gene promotes the transcription of this gene. This is self-activating. There's some feedback, some repressive loops and so forth. And you can build these, I can't, bioengineers can build little genetic circuits and put them inside bacterium. Okay? And tag them before they put in with a blue or a yellow or a green fluorescent tag so that they can watch the variation through time of the sets of genes that live in a little biochemical circuit that they designed that looks like one of the mammalian circuits that is interesting, for example, in cancer. So that's the context. That's one of the reasons we're interested in. So what do we have here? Statistically, we have lots of things, lots of data problems. Fundamentally, we want to get down to the level of modeling. I'm going to show you some models of little nonlinear dynamic multivariate time series, which is what you get out of this stuff, where the data comes in the form of these really fuzzy movies and is very limited by imaging technologies, by microscope technology currently, as well as the ability to measure more than a few genes, a few RNAs simultaneously. So that's where we are in terms of the technology. Where we are in terms of statistics, we know a lot about how to fit models, of course, and the one sort of technical statistical thing here has to do with fitting hidden Markov models. I'll give you a couple of slides on that. Because we want to fit models that have population parameters that represent various rates of interactions, but then allow each cell to be different in terms of the levels of the RNAs on the sets of proteins that are being monitored through time, and that allow stochastic components that allow you to soak up anything that isn't modeled in there and to begin to introspect as to what that might mean in terms of model misspecification. Making comparisons across different cells. Nobody has done this yet, but it's part of where we're going in our projects. Very, very active right now is the idea of fitting models and then slightly changing the engineered circuit that's been put inside the bacteria to put in a little feedback loop or a repressive loop or take something out, predict from the model what you expect to see, and then go ahead and do it and generate the experimental data. So here's a set of genes. The little black one is something we control in such a synthetic circuit. There are 3 genes, red, green, and blue here, and they interact in various ways biochemically, and we know that's true because my colleagues in biochemistry and bioengineering designed it. And that then gives us a three-dimensional state variable at each time t, the level of RNA of those genes. So x is state, and we make measurements, and the biochemical models say, well, there's going to be a change in small time intervals that's stochastic, and there's some activation, repression combination of such terms that come into a state space equation, typically very nonlinear. So here's an example in this little system. The first element is basically decay of the protein labeled Ly, of the RNA of the protein, it doesn't matter, of the first gene. The second element has a nonlinear activation, a nonlinear response function with some parameters in it, some decay terms. So there's 7 or 8, maybe 12 parameters in there. And then there's some parameters that characterize the noise. The measurement error in the first equation, the errors of measurement in actually getting the data. You don't actually see intensity of RNA, you see level of fluorescence. And there's a series of machines that give you that data. And so we're just going to talk about what we do there, normal distributions, compound gamma distributions, slightly non-parametric versions are possible. And then from the movies, we get the data, which are the y's at each time t, and these are actually data just on one of these genes, T7 in this circuit. Some of them just sort of wobble around and decay, some wobble around and then experience a region in the dish that they're living in that is serum stimulated rich and start growing, and some go down and then go up. So you see the individual cell trajectories over time here. Okay? This is data we've reconstructed from the images, and there's about 40 snapshots in time, at about 40 time slices. You take a photograph with a camera, then you have to wait a little while before you can take the next one, because there's a delay in the registering the fluorescence that screws up the observations if you take them too close together. So the models live on a defined time scale, but the data is on a cruder time scale. So we're in the business of imputing what is missing, the latent states that are missing. Okay, so to recap, many cells, you saw the bacteria evolve from a few, 50 or so to begin with, to several hundred. All sort of mashed up and obscuring each other. So there's multiple cells, and we're measuring a small number of RNAs in each cell. There are some population parameters, and then there are the levels of the proteins, the RNAs, corresponding to the genes in the model for each of those cells over time. At particular time points, you make a measurement with noise, but for most of it, you don't, because the model lives on a much finer time scale. So what we do, and the one technical innovation I'm going to talk about, is in an MCMC component for dynamic nonlinear models generally, but it was motivated by these applications. Markov chain Monte Carlo methods, of course, where we iterate between simulating parameters, a handful of parameters, or 12 in this example, and then conditional on the parameters, we find approximations to posterior distributions to simulate the latent states for all of the cells over time. Just as a side note, in case there are people in the audience who live in the biological world or work with biologists, there are other technologies to bring data to this kind of problem. You can grow batches of cells labeled like this in 50 trays, in 50 dishes, and then after a minute you can process the first, and after 10 minutes you can process the second, and get very many observations, thousands and tens and hundreds of thousands of cells at a given set of time points that provide sort of marginal data from methods called flow cytometry. I'm not talking about that today. It's data that doesn't give you time series because it doesn't match up the cells. Okay? But it's additional, very high dimensional data. And if you don't know about flow cytometry and you work in biology, you will know about flow cytometry. It's becoming ever more present, and it generates massive data. Okay, so back to the nonlinear state space imputation question. So here we are moving through time. This could be in a talk in economics. It's not specific to systems biology. So here are our states at time t plus 1, and so on and so forth, time t plus k. And at this time point, we make a measurement on the state. At this time point, we make a measurement on the state. But the model lives on this finer time scale, and so we're skipping k minus 1 time points. The model is then going to help us think about what the trajectory of the states across that interval, and as we move through time, future intervals really is. It's a hidden Markov model. The nonlinear state space model is a Markovian model. It's hidden because we don't make direct measurements. They're always noisy. And the very common, the very standard idea then to build a method of simulating posterior distributions of hidden states is what's been called the forward filtering backward sampling method. It's a sort of Viterbi style simulation method where you process data through time sequentially, moving from the left to the right, and at each time point you have what you currently think about the current state given the data up until that time point. So here the dt, the data would include yt. At this time point, the dt plus 1 wouldn't have any new data. It would just have yt in the previous data. So forward filtering, if you know about this kind of stuff, Kalman filtering, basically recursively squares, works in linear Gaussian systems. And if you're outside that framework, then nothing works analytically. You have to do some kind of numerical work. Okay? So what we want to do is simulate. We go through calculating distributions like this, then we get to the end, wherever the end may be, and we have a distribution at the end, so big T means the end, and the backward sampling or reverse sampling component of the so-called FFBS kind of analysis begins by sampling that distribution to draw from the posterior distribution of the final state, and then recurse backwards in time using the Markovian structure of the model, simulating from distributions like this, simulating a value of the state here, conditional on the simulated value of all of the states that would follow it, because you're running backwards in time. And because of the Markovian structure, that comes from the joint distribution of consecutive states. So fundamentally, there's a focus on pairs of consecutive states at the heart of wanting to do this kind of calculation. Jared Nemi, one of my students, is finishing up his second paper on doing this with an old idea. And when he came to me about a year ago and said, this is the way we can do this, I said, oh, I published a paper 15 years ago on precisely that. Didn't I give you that paper? And he said, yeah, but you didn't quite get it right. And he is absolutely spot on, absolutely spot on. I didn't kick myself because it's great, he's a great student. But the basic idea is very simple. Let's pretend we're doing the filtering, and this is our model, and this, whatever it is, there's some nonlinear function here. We're moving forward in time, and we currently have a distribution for the current state given the data at that time that happens to be a mixture of Gaussians. There it is. Okay? Well, it's a mixture of Gaussians, and it's a mixture of a lot of them. It's in 3 dimensions, of course, in our model. And they're all very tight. It's a kernel density estimate, if you like. Okay? A mixture of very many Gaussians. So each of them says that X is in a very small region in state space. Okay? Here's a particular nonlinear function G, okay, in this state space equation, just as an example. And on the range of variation I've plotted, each of these little ellipses is one of these Gaussians, okay, down here. So they're very tight. And so if you project that onto the nonlinear function, you're very close to being, you're so confined that, you can fit a straight line in there. It's not going to matter because the ellipse is confining you, okay? And that's a sort of old idea. Was used actually in the early 60s in both statistics and engineering literatures. Almost, not quite. So the basic idea is then if you start off with a mixture of Gaussians and use some trickery here, you get a mixture of Gaussians, and everything then flows. You can do the forward filtering, you can do the backwards simulation, okay, because if you can do calculations with Gaussians, you can do calculations with mixtures of Gaussians, even if there are very, very many, in a simulation-based analysis, okay? So it's really not, almost non-parametric approximation of nonlinear state space models, okay? Here's another example. You can't quite see that, but there's a red dotted line running through those contours. You can see it's wiggly. And here's a true joint distribution in a particular example with a 2-dimensional xt plus 1. And there is such a bivariate mixture approximation. Okay? Now, I put this up not to kill the point, but because you can see from this frame that there seems to be more spread. Okay? And that's by design. Okay? Because 15 years ago we could have done the calculations I've just described, but 15 years ago we wouldn't have been able to exactly simulate from the exact posterior distribution of the whole sequence of states. Now we can. And the additional component is that we don't use this as the model, we use this as a Metropolis-Hastings proposal distribution to generate sequences of states from this mixture modeling idea that are then tested and either accepted or rejected, and if they're accepted, they're draws from the posterior distribution in the exact nonlinear model. So the idea is to use good old, what people used to call mixtures of Kalman filters or Gaussian sums as Metropolis-Hastings candidates. And in a series of examples this works. So the idea is here are two cells plotted on top of each other over time, two of the real data sets. You can't see the dots, there are black dots in there. But you run this over and over again, and each of these has three trajectories. Each of these has three simulated x's overlaying the data. Okay, so we get posterior samples from the posterior distribution of full trajectories. And in that data set with 40 observations, we've got about 1,000 time points that we're doing this, we're simulating over. This is one little model. It's not so much of the interest here is biochemist A gives you this model, biochemist B gives you this model. They're different models. One has a feedback loop, the other one doesn't. Okay? Or they have different interactions of proteins coming together to form complexes that are required to perform a function. Okay? So it's all about model exploration, evaluating models by fitting them and making comparisons in various ways. And this is one simple model that is a bit more elaborate than the other one. It's got a background noise term in for cell C at time t. And this is a one-dimensional state where there are 5 parameters coming in here. And so you couple that simulator of latent states with simulations of parameters given the states to generate posterior distributions for the model parameters. And right now, a big part of that is trying to figure out, in this class of models, is there any opportunity for this kind of data to tell us anything about these parameters? Okay? And what we have here is posterior histograms with prior densities overlaid, where the story is very clear in some cases. There is useful, informative information. The data is telling you something. It's giving you a posterior that's very different from the prior. But with other parameters, it's pretty clear. And then you go back and look at the models and say, well, I'm going to need much finer timescale data to see those effects in the first place. Perfect. So it is, it's perfect. I've got lots of movies of different kinds of cells, but this is the same bacteria. I just want to say two more words on data. Okay? Slowed it down, taken away a few of the cells, and we've colored white. It was one cell. You can probably, well, those at the front can see it. There's 2 cells now. It's about to be 3 cells. Now it's 3 cells. Now it's 4 cells. Okay? So we've outlined, we've registered an initial cell there, and we're following it through time. And when it goes through mitosis, it's got 2 daughters, and we're following the daughters through time. Okay? So this is yeast, this is E. coli, we've got yeast, and we've got mammalian cells. So started this project, got into this area about 18 months ago, and we're ahead of technology. Statistics is way ahead of technology. There'd been a couple of papers, one Science and one Nature paper, and we started working with those 2 groups because we wanted their imaging methods so we could start getting data out of our movies. And it turns out that you don't just need their imaging code, but you need at least 3 of their lab techs and postdocs plugged into it. Okay? There's lots of imaging stuff. Okay? The world is full of imaging in the public domain, in public domain software, and also lots of very, very expensive, fancy commercial stuff. We've tested them all, and we've had everybody who's expert in them test them. And then after 6 months, we gave up because it wasn't working, and we developed our own basically as a sidetrack on this project, because we have to do this on a series of frames over time. And then we have to be able to say, well, this was the cell at frame 1. This is where it was at frame 2. We're putting a box around it because we want to measure intensities of the stuff that cell is throwing out. And at frame 3, it was here and here. It went through a cell division, okay, and there are its daughters, okay? We want to do this routinely. And it's much easier with bacteria than with cancer stem cells, any kind of mammalian cell. And the software, the tools just don't exist. So one of our group developed, took 12 months, but it was worth it, developed a piece of software that now chews up movies of basically arbitrary objects, segments and links. Quanley Wang is the imager, to present this data. If you're interested in any of this, there are various papers on the imaging and the time series on the website here. But again, as I said in opening, we're all used to high throughput biological data, molecular data, genomic-y stuff, okay? This stuff is not high throughput. The models are high dimensional, okay? But this whole field is getting to the point where it feels to me that we're about where we are now with this field as we were with microarrays 10 years ago. Okay, most of you didn't know about microarrays 10 years ago. I learned about them 9 years ago. Okay, and the technology is moving. I don't know what the technology is, but it's moving ahead so much that I know in a very short time, a small number of years, we'll be able to look at movies like this where we're measuring 50 or 100 or 1,000 RNAs in real time. And the whole business of understanding genetic architecture and cell-specific effects, all of the important stuff when it comes to human diseases that we can't look at nowadays, that's all coming, okay? And data is going to be really big. Anyway, thank you. Thank you for your attention.
I'm quite sure how strict we are for time, but if there's a burning question, perhaps we could have one.
What kind of resolution do you have? Can you trace well the exact location of the protein at what different?
No, this is the unit of resolution is the cell here. We care about an aggregate measure of the level of expression of that gene at a given time. Okay, we're not actually looking at spatial location. In other studies, I'm not involved in them, but I know it's possible to localize individual proteins within a tissue. There's a lot of that kind of work in embryonic research, for example, when cells are differentiating and moving around. Okay, so there's a lot of very useful imaging technology, but here it's the labeling technology that's limiting, the ability to measure more than several.
Okay, let's thank Mike again.